Now that we've reviewed what exactly is gene expression, going from DNA to RNA to protein, we will now look in more detail at the process of transcription itself, going from DNA to RNA. We'll begin by looking at the organization of a gene, what actually constitutes a gene, what percent of the genome actually is protein coding regions, and then we'll talk about the stages of transcription, which include initiation, elongation, and termination. So when we are looking at a genome, so thinking about that double-stranded DNA, what we want to focus on now is the regions that actually get transcribed and ultimately translated into proteins. And if we were to examine the DNA, only about 1% of your genome is actually protein coding. And so something like 99% of our genome actually is not involved in ultimately becoming a protein. For a while, this was actually called our junk DNA, but we now know that that is indeed not the case. And a large portion of what this quote-unquote junk region is its function is to actually regulate when a particular gene will be transcribed. And so how do we know what region is important for that? So the region just before the gene, or just upstream of the gene, we call the promoter region. And I'll explain what the function of that is, but really all the machinery is going to assemble there, and then that's necessary for transcription. And then we have regulatory sequences all throughout the genome. And I'll talk a little bit more about that as well. But the regulatory sequences will assemble particular types of proteins called transcription factors, and that will drive the levels of expression of a particular gene. Just downstream of our transcribed region, we have what we call the terminator sequence, and this will be the signal for all the machinery to kind of dissociate from the DNA, as well as the newly transcribed mRNA, and then the mRNA can be processed. So a couple of key regions again to point out is this promoter region. So here is where the main enzyme, which is going to be RNA polymerase, the name again tells you what it's going to do. It's going to polymerize a new strand of RNA. That's where the RNA polymerase is going to bind, and that's where transcription will initiate. This regulatory sequence, as I mentioned, this is going to be the main site for our transcription factors, regulatory proteins, and so what the regulatory sequence does is it will stabilize or destabilize the binding of RNA polymerase at the promoter. So the tighter that RNA polymerase is bound to the promoter, the more RNA that you will ultimately get. So this regulatory sequence facilitates either tight binding or loose binding of RNA polymerase and will really regulate the amount of transcript that is ultimately made. Again, our transcribed region, this is going to be what ultimately gets made into protein. And then again, that terminator sequence is where the whole, all the machinery is going to disassemble and fall off of the DNA. And then the DNA will just come back together. That double-stranded region will come back together and form the complementary base pairing. All right, here's showing that region in a little bit more detail. So again, in purple is our RNA coding region that ultimately contains the portion of the RNA that will be made into protein. So just upstream, we call this upstream, just before that RNA coding region is our promoter. Again, that's where RNA polymerase is going to bind. Upstream of that is our regulatory sequence. That's going to be the primary region where transcription factors can bind. It's certainly not limited to just upstream of the promoter. We can find regulatory sequences actually all throughout the genome, and I'll talk a little bit more about that. And then again, downstream of our RNA coding region is where we're going to have our terminator, and that's where RNA polymerase will dissociate from the gene itself. Okay, so when we're talking about transcription, we've got these three key steps: initiation of the process of transcription, elongation, and termination. So initiation is incredibly important. Again, this is when RNA polymerase is going to be binding in that promoter region where all those transcription factors are going to be assembling in the regulatory region and stabilizing the binding of RNA polymerase. And then that's where transcription will begin, is at that transcription start site. Elongation is what's going to happen when RNA polymerase now makes its way down the gene itself, and we're going to transcribe that new RNA molecule going from DNA to RNA. And then termination again is where the RNA polymerase will disassemble, as well as the newly transcribed RNA and the double-stranded DNA will come back together. The promoter region, as I've stated, again, is where RNA polymerase is going to bind. Those transcription factors, they are going to interact directly with RNA polymerase, and their main place of binding is going to be in the regulatory region, just upstream of the promoter. However, I want to point out they can also bind in the promoter. They can bind upstream or downstream of the gene. I studied transcription factors in graduate school, and the binding site for the transcription factor I was looking at was 22,000 base pairs away from the actual promoter region. And so I don't want you to think that it just has to be adjacent to the gene. It can actually be quite far away and still direct the expression of that particular gene. And then again, RNA polymerase is going to be what is the enzyme that actually is going to synthesize our new RNA, the new RNA molecule that we're making. And so it has a function similar to DNA polymerase, but it actually has capabilities sort of like our helicase as well. So RNA polymerase is going to be able to unwind the DNA and then go ahead and polymerize a new strand, help to polymerize a new strand of RNA. And it is going to move, just like DNA polymerase did, in the 5' to 3' direction. All right, so let's look at the actual process of initiation of transcription. So again, the key is that in the promoter region, we have RNA polymerase that is going to bind in that promoter. And if we're looking at bacteria, they have something called a sigma factor. And sigma factor is important for binding. It will bind to the promoter region and stabilize the binding of RNA polymerase, sort of like transcription factors. And as it approaches that transcription start site, again, the RNA polymerase is going to begin to unwind that double helix so that we have some single-stranded DNA to act as our template for the RNA strand that is going to be made. So once we've initiated our complex and we begin to move down the gene towards that transcription start site, and the double-strand DNA is going to be unwound, the RNA polymerase is going to begin to facilitate the polymerization of that new strand of mRNA. That sigma factor, this is true again only for bacteria, is going to dissociate and is no longer needed. And RNA polymerase will just make its way down the transcript itself. Again, we are using just one of these two strands of DNA as our template, and we are going to be moving down the DNA, synthesizing the new RNA strand in the 5' to 3' direction. Now, we've talked a lot about the differences between DNA and RNA. As we are forming the complement, rather than having thymine in RNA, we've got uracil. But other than that, all the complementary base pairing stays the same. Eventually, a terminator sequence will be reached, at which point we will have disassembly of this whole complex. The RNA polymerase will dissociate. The newly formed mRNA is going to also dissociate from the double-stranded DNA, and the double-stranded DNA can now reanneal and form the complementary base pairing. Some additional terminology that you'll want to make sure you have a handle on is being able to differentiate on the DNA between our coding strand and the template strand. So the coding strand is what our mRNA transcript is going to look most similar to. So if you look at the the sequence of this strand highlighted in red. You see it has an ATG, GAG, GAA. And now if you look at the transcript, it's going to be identical to the coding strand with the exception of T's have been replaced with U's. So our transcript looks like our coding strand. However, if you recall what RNA polymerase is doing, which is causing those two strands to pull apart, the strand that the mRNA makes a complement to is actually our template strand. So when TACC is what is revealed, the complementary base pairing that takes place between the DNA and the RNA temporarily is going to be between the template strand and then this primary transcript, the RNA. So the T, as always, is going to complementary base pair with A. A rather than with T is going to bind with U. A C is going to bind with G, etc. So again, just to reiterate one more time, our primary transcript is going to look like our coding strand on the DNA. However, we used the template strand to form the complement. All right, so this will kind of address whether or not that just made sense to you. So I want you to answer the following question. Which of these two strands is the template for transcription? So go ahead and pause the video and answer this question. So hopefully you answered that indeed B, the blue strand, is our template strand, and I will explain why. So if this is our promoter, then again RNA polymerase is going to bind here upstream of our gene and then transcribe and unwind that DNA and allow for the new RNA strand to be synthesized as it's moving down this direction, and that RNA polymerase is going to be polymerizing a strand in the 5' to 3' direction. So if it's going to form a new strand in the 5' to 3' direction, it means we need to be working off of the complementary strand, which is going to be in the 3' to 5' direction. So this is going to be our template strand. The blue strand will be transcribing RNA in the 5' to 3'. And what is that new RNA strand going to look like? It's going to look like the red strand, the coding strand. Okay. And just to show you some of the complexity of the natural genome, really the important thing is to identify the promoter region, because again, the promoter is where the RNA polymerase is going to bind. So if you know where your promoter is, RNA polymerase will bind there, and then you're going to move down that gene in the 5' to 3' direction in terms of RNA synthesis. So for this particular gene, gene A, the RNA polymerase will bind here in this light blue region, will move down the gene, and will be transcribing in the 5' to 3' direction. So in that situation, our template strand is going to be this bottom strand. Again, if this is our promoter for gene B, and we move and transcribe in the 5' to 3' direction, we're going to be using that bottom strand as our template strand. However, for gene C, the promoter is actually shown here, and if we're going to transcribe in the 5' to 3' direction, in this scenario, the top strand can actually become our template strand. So it's really important to identify what's your actual promoter region, where's your transcription start site, where's your terminator, and then from that you can figure out which strand, either the top strand or the bottom strand, is going to be your template. All right, so I want you to take a second and determine what is going to be your transcript. So if this is your DNA and the promoter region is here and we're transcribing in this direction, what is going to be the new mRNA strand that you synthesize? So go ahead and pause the video and write that out and give yourself a second to see if you're right. All right, so this bottom strand is going to be our template strand, because again, we need to synthesize in the 5' to 3' direction. So we're going to form a complement with the strand that's in the 3' to 5', the opposite. So hopefully you came up with something like this. Our new RNA product is going to be ACG, forming a complement here, AUGC. And if you notice, this looks identical to our coding strand with the exception of the T's have been replaced with U's. All right, so that is basically section 10.2. That's how genes are organized, the stages of transcription, initiation, elongation, and termination, and you should be able to describe those thoroughly